So thank you for everybody joining the Marine Institute Graduates Society seminar series. As we've said before, you can find some of our previous seminars on our YouTube channel. And before we start, I want to provide a land acknowledgement. So we respectfully acknowledge the land in which we gather as the ancestral homes of the Beothuk and the island of Newfoundland as the unceded traditional territory of the Beothuk and the Mi'kmaq. We would also like to recognize the Inuit of Nunatsiavut and Nunatsiavut and the Innu of Nitassinan and their ancestors as the original peoples of Labrador. We strive for respectful relations with all peoples of this province as we search for collective healing and true reconciliation and honor this beautiful land together. So today we have Dr. David Cote. David Cote is originally from Kitchener, Ontario, and found his calling amidst the natural splendor of Terra Nova National Park in Newfoundland, where he began his Ph.D. exploring the habitat and movement ecology of juvenile Atlantic cod. Transitioning to a role as an ecologist with Parks Canada, David shaped the aquatic ecosystem management of several national parks, fostering an appreciation for the delicate balance between protected areas, adjacent landscapes, and local communities. Today, as a research scientist at Fisheries and Oceans Canada, David leads a team of devoted to helping Canada achieve its marine conservation targets in Newfoundland and Labrador. Collaborating closely with Indigenous communities, harvesters, academic institutions, his team harnesses the interdisciplinary synergy to characterize and safeguard the region's marine biodiversity. Today, David will be sharing his experiences on the challenges and opportunities related to creating and protecting the rich biodiversity of some of Canada's most beautiful areas. And with that, I will pass it off to you, David. Thank you.
Thanks, Gord, and thanks everyone for attending this talk today. I guess it was challenging to come up with a talk because we're spread across so many different aspects of the ecosystem that it's hard to thread something together. And I thought that maybe talking about the program in general and some of the challenges and opportunities we have would be useful and informative of just the kind of things that we're doing. But this really only touches on some of the things that we're at. First of all, I'd just like to give you a bit of information about who we are. And we call our team SIREN, which stands for Conservation, Integrated Research and Exploration in Newfoundland and Labrador. And our main mandate is to provide science that supports Canada's Marine Conservation Targets program. So we've been in place since 2017, and we work closely with Indigenous, academic, industry, NGO, and government partners. And given that this talk is at MI, I'd like to acknowledge all the important contributions that MI has provided to our program over the years. And a lot of the work that we're talking about today has been done in collaboration with MI. So just want to touch on protected areas in general. And so as Gord mentioned earlier, I started my career in Parks Canada. And that was, I guess, about 25 years ago that I moved out to Newfoundland. And even in that time frame, I've seen quite a bit of change in terms of how protected areas are managed and incorporated into our society. But we can go back even farther, and you can see back in 1885, Parks Canada initiated its first protected area in Banff, and it started out pretty much as a spa. So they found these hot springs at the end of the rail line that connected the entire country together, and they thought that would be a great reason for people to travel the trains and go out to Banff and enjoy some time in the hot springs. And so since that time, things have changed quite a bit. You know, we're focused away from spas and golf courses to more biodiversity conservation. The way we set up protected areas is less about expropriations and more about consultation. And we've changed from protecting parks and protected areas from people to protecting them with people. So in many cases, we see Indigenous groups are allowed to use these protected areas to continue their cultural practices. And then things like laissez-faire management versus active management, or universal policy versus co-management systems are much more prevalent. And so in general, we're seeing a much greater emphasis on social, cultural, and economic outcomes in addition to those conservation outcomes that are happening. So in the marine realm, we're quite a bit farther behind in the terrestrial realm. And so the first national parks, or marine parks, sorry, started showing up in Canada in the 2000s, over 100 years after the first terrestrial park. And so since then, things have been moving really quickly. And Canada recently signed on to an international agreement to protect 30% of its oceans by 2030. So that's a huge, huge challenge for us to achieve. So right now, we're at about 15%. And if you look at the top plot, I don't know if you can see my cursor, but this top plot up here shows the existing protected areas, which represents about 15% of our ocean area. The Newfoundland and Labrador bioregion that we're in here is shown with the red polygon. And if we look at the size of these areas, we can see they range from the very small. So Eastport Marine Protected Area is probably one of the smallest. It's the size that if you blink, you probably would miss it as you're driving through with a boat, to very large ones that are greater than 100,000 square kilometers. And if we look at the depth profiles, we can see that they're heavily skewed towards the deeper environments. And I just popped up a rectangle there to show the depths that are typical for the operational limits for our research vessel trawl surveys. And you can see that we have a lot of depths that fall outside that range, which makes it really challenging to use existing monitoring to do it. So in the Newfoundland bioregion, we have some incredible data sets derived from our research vessel trawl surveys. And you can see each of those yellow dots represents a trawl set that's been done over the past decades to collect the data sets that are used to manage fisheries. And so really valuable data sets. They're economically, they drive economically important industries. But we have huge regions shown here in red that are they're not sampled at all, and we know very little about. And so when we first started in 2017, there was a big emphasis to start looking at the Labrador deep ocean, and a little bit later in coastal Labrador. And it was a bit of a daunting task because in these areas we have, they're very remote, they're prone to unpredictable weather, they're ice covered for much of the year. And we also had very limited equipment to sample these areas. So the trawlers that we have in Canada typically can reach down to about 1,500 meters if we're stretching it. And we needed to go well beyond those depths. And we also had a lot, not very much institutional knowledge left about sampling deep sea environments. And so that was another challenge that we had to overcome. So our approach to trying to figure out how we characterize these huge areas with a small team and a limited budget really relied on trying to understand how biodiversity changed across environmental gradients. And so this was one of the first programs we initiated called Ice Cold Program. And basically what we did is set up 4 transects that went from the shelf break out to the deep ocean, 2,500 or 3,000 meters, depending on the transect. And across these transects, we would do a whole bunch of different operations, everything from, pelagic netting to drop camera work and, water sampling, etc. And so we thought we would get a lot of information, but I'd just like to point out how difficult it is working in these environments. It takes so long to get a sample from 3,000 meters that you can't realistically do very much sampling. And so we knew that our program would have trouble building the data sets we'd need very quickly without thinking carefully about how we did it. And so a big part of what we did was try to build on existing data sets and knowledge where we could. And so one example of this when we were looking at demersal environments was a long line program. And so long lines, you can see the picture in the bottom is a pound of rope here where the rope is streaming out the back and all the hooks that are attached to this line go out. And at the deepest depths, there would be about 13 kilometers of rope that would have to be shot out of the stern of the fishing vessel and then also hauled back in later on once we had set it out for our 12 hours. The great thing about this method, though, is that we had corresponding data that had been collected a couple of decades earlier around the Flemish Cap, you can see with the yellow triangles. And so we could compare what we found up in the Labrador Sea with the red, where the red dots are, to the Flemish Cap and get some idea of if what we were seeing was very different or similar across these areas. And so what we found when we plotted both of these regions in terms of species richness, biomass, and abundance was that both of these communities varied a lot with depth, and that's not surprising. But what we found in each region was that these metrics were the greatest at these intermediate depths of around 1,500 meters. And importantly, there were very similar patterns across the region. So that allowed us to infer what was going on in the 1,200 kilometers that separated these two regions, and gave us a much better idea of what was happening out there, and which was particularly useful for, what's trying to assess the potential impacts of oil and gas and other activities. So in a similar manner, we also did some baited camera work in these habitats. And again, the Labrador Sea is circled in red, but we also were able to get our hands on data sets from around the Atlantic, including the Mid-Atlantic Ridge, the Northeast Atlantic, and also some of the Southern Hemisphere. And what you're seeing there and this area here is a multivariate plot. And the closer 2 points are, tells you how similar the fish communities are from these baited camera sets. And so we can see the Labrador points here in light green, and clearly they're to one end of this cloud of points, showing that they're kind of distinct. But what's interesting is when we look at all the deep water communities across all these regions, you can see that they all aggregate in one area. And so those are represented by the squares and the plus signs and the open squares with X's in them. And so even though there's huge areas across the Atlantic, there are parts of our ecosystem in the Labrador Sea that are very similar to other areas in the deep water. Now, if we contrast this to what we've got in shallower shelf environments that we did later on, we can see that they're very different communities. In fact, there's hardly any overlap whatsoever with what we're seeing at the continental shelf break. And the shelf communities of the Labrador Sea are very species poor communities, where we have species like rock cod and different types of sculpins. So collectively, these two studies give us a really good indication of what makes the Labrador Sea special and what makes it very similar to other regions. And we can do similar things that duplicated there. Oh, before I move on to the mesopelagic zone, I just wanted to show you a little video of what we see down at 3,000 meters. And so 3 kilometers below the surface, most people assume there's not going to be much there. But in fact, even in those depths, there are pretty vibrant fish communities that we might want to protect. And these are all abyssal grenadier, these ghost-like fish that we find characteristic at depths beyond 2,000 meters. And okay, so if we move on to the pelagic zones, while we're doing those transects, we're collecting a bunch of data in the mesopelagic zone. And as good as DFO does at collecting data from demersal habitats along the shelf, we probably do a lot less work in the water column. And so on these transects that we were doing on the Amundsen, we were able to collect a lot of data on the mesopelagic zone with hydroacoustics and with pelagic nets. And this was done with in collaboration with Max Jeffroy's lab. So if we look at these panels on the left, we were able to run these fisheries acoustics devices from the Labrador Sea north into Baffin Bay. And so you can see in the map for this left panel up here in the Labrador Sea into Baffin Bay. And as we traveled from the south to north, we could see these very distinct deep scattering layers which are well known around the world for areas of aggregated pelagic fish and plankton. And they aggregate around that 500 meter mark. And you can even see evidence of them doing some diel vertical migrations in some cases. But as we move north across the polar front, where we're moving into the really cold waters of Baffin Bay, and you can see depicted by this red line, all of a sudden, this deep scattering layer disappears, largely disappears from the hydroacoustics. And what's interesting is when we compare it to a corresponding data set in the Southern Ocean, we found the exact same thing happen. If we were moving north, or sorry, from the north to south, so from warmer water to colder water, the same thing happens, a very distinct drop off as we cross the polar front in these mesopelagic habitats. So the prevailing theory was that these communities don't exist in the polar areas, largely because there's not enough light in these areas. But because we found these sharp drop-offs at polar fronts and that they occurred at different latitudes, suggests that water temperature is a really important driver in these pelagic communities. And that has important implications because light levels are not going to change over time, over decades, but water masses may change with climate. And so we could see shifts in whole communities based on this. And if I can draw your attention to the right panel, we did similar things with mesopelagic netting. And so these are plankton nets that go to multiple depths, and they're the exact same nets that were used across the Arctic, across a whole variety of samples. And so if we look at the Labrador Sea samples, you can see they're depicted in a different color of yellow. These are the offshore samples, and they're completely distinct from everywhere else in the Arctic. And they're also distinct from the coastal samples that were taken, which are much more characteristic of Arctic type communities that we see in shallow environments. And so again, another example of taking a limited number of samples in the Labrador Sea and being able to leverage it into much greater context to understand, to characterize these areas better. So after several years of working in the deep ocean, we were asked to put a bit more attention in coastal Labrador because there are some protected area initiatives that were underway there. And so the interesting thing about coastal Labrador is we still have very limited scientific information there, but we have the benefit of very rich Indigenous knowledge. And so Inuit have been living along these coasts for thousands of years. On the top, you can see a picture of some tent rings of where people would return to harvest marine resources. This is up in Nukvuk Fjord. And if we overlay what we do know about coastal Labrador next to Nunatsiavut's land claim zone, the dots here represent our trawl data, and the red line represents the land claim zone in the southern part that we are doing a characterization for. And you can see that they're almost mutually exclusive, that we had almost no information in these habitats. Fortunately, local knowledge from Indigenous people were able to fill in a lot of these gaps. And so here we can see some maps that were created based on local knowledge, and you can see the shaded areas of yellow and purple represent the knowledge of the distribution of Atlantic cod. And the interesting thing about it along the coast is where climate fluctuates and species distributions shift, we get some knowledge that extends across multiple decades. And so we may not necessarily see Atlantic cod up in the northern reaches every year, but because people have lived in that area for so long, we can get that type of information. And so all this information was compiled together in a Canadian Science Advisory Secretariat report for coastal Labrador. And this is a DFO process, but it was the first one ever to include Indigenous knowledge and Western science on equal footing. And so that was a big milestone for it. And we feel that we had a much more comprehensive document with which that we could move forward for proposing protected areas. And so in addition to characterizing these areas, we also have a lot of research to do. And again, there's been a real emphasis around the world on inclusion of Indigenous groups when we're trying to research protected areas and implement them. And so, and Canada buys into that, and accordingly, we've been working in collaboration with Nunatsiavut whenever we're working along the coast. And so that involves incorporating local priorities into characterization work. And so Nunatsiavut has their own research program, and in the marine environment, it's called Imapavut, which means our oceans in Inuktitut. And that corresponds well with our coastal characterization program we call ICICLE. And one example of that is we worked with Oceana, which is an NGO, and Nunatsiavut to identify some important areas to sample for some benthic characterization. And local people directed us to sample some areas close to culturally important sites. And you can see the fjords of Hebron and Okak Fjord. Those are two areas of resettled communities that were really important to local people. And of course, areas around Nain, which people inhabit right now. And we saw all sorts of interesting animals and creatures while we did this. Interestingly, some of the highest biodiversity that we found was located in polynyas, which were listed as culturally important hunting areas. And I'll just show you a little video of some other things that we found up in Hebron Fjord. These are Serianthid fields, which I love to look at because they kind of remind me of the Avatar movie. But some really beautiful creatures in these environments that we were able to find based on direction from the Nunatsiavut. All right. So just to close off this, some of this work in coastal Labrador, just last week, Nunatsiavut and the federal government jointly announced the Torngat Marine Conservation Area. And this is an area of interest that is adjacent to Torngat Mountains National Park, and it's going to be protecting fjords and shelf habitats of northern Labrador. So a lot of the work that we put forward and we helped analyze is helping characterize this particular area. And so we're pretty excited about that. All right, so spent a lot of time talking about characterizing areas and trying to get the general essence of particular habitats. But there's also a need for finding special areas as well, areas of particularly high conservation value. And so I always like to go back to, the origins of deep ocean research. And, in that same era when Banff National Park was being created, we are just starting to do the very first deep ocean surveys. And so the Challenger expedition that started in 1872 was basically a converted naval vessel, a wooden vessel, and they sailed around the world for 2 years, sampling deep sea environments all over the place. They never made it up to places like Labrador, but they did cover a lot of ground. And they were using tools like you see here on the left, which is a rock dredge. And they had to do, process these samples, digging through them, the monotony of digging through mud to find little critters. And so not much has changed in many respects because we have the same tools. You can see the left rock dredge that we've used on the Amundsen. This is our research vessel, which is probably a little bit more comfortable than what they had. And we also have scientists that have to go through the monotony of picking through sediment samples, which take a long time. Now, the Amundsen has some extra tools on board, which we're lucky to have, including hydroacoustics that I mentioned earlier, but also some, drop camera imaging systems and things like ROVs. And all that comes in really handy for, different ways of exploring the ocean depths. So typically when we leave the wharf looking for these areas of high conservation value, we're hoping to see areas of really dense biodiversity and, sensitive species like corals and sponges and weird and wonderful animals like the umbrella octopus you see in the bottom right. And inevitably, if we don't go around with informed site selection, kind of like the Challenger had to do, we end up with mud samples. And so there's lots of virtues of mud. It does important ecosystem services like carbon sequestration, and it has its own set of animals. But there's an awful lot of it. And so if you close your eyes and pick a point on the map in the ocean, you're likely to probably put your finger on a place that's covered in mud. So to find some other types of habitats that are a bit more rare, we need to use all the tools available to us. And so one easily accessible tool is GEBCO bathymetry. And you can see that's a pretty blurry image, but you can kind of make out some areas where there's some bathymetric relief, areas where sediments may not accumulate, and you might have some hard bottoms that would support different communities of epifauna. And then similarly, we can use bycatch data. And this again is from trawl and fishing fleets. And this is from a paper by Lauren Gullidge. And we can see the big circles represent areas of high densities of sponges and corals. And so that might give us an indication, but of course, the fishing surveys are limited in terms of their spatial extent, and they're not much use for us in coastal Labrador or in the deep ocean. But with these two data sources combined, we can also do things like habitat suitability modeling. And again, this is the same paper by Lauren Gullidge and an example of a model for large gorgonians. And the black dots represent areas where they were captured, but the other red areas represent high suitability habitats. And you can see that the vast majority of these habitats occur on the continental shelf break, areas that we already assume that they're dominating. But I want to draw your attention to an area in the Makovic Trough area. There's an incursion of deep water, and we can see a few flecks of red and yellow of data there, which implies that there's potentially high suitability habitat there. But it's a huge area, and really not great for the tools that we have, like ROVs, that kind of require precise coordinates. So we started scouring our available information, and we have some bit of a treasure trove of information from a retired fishing captain named Wilford Bartlett, who used to fish on the Labrador coast. And he attended a meeting that one of our biologists was there, Vonda Hayes, and he told her about this bycatch of corals that he was getting. And while he didn't have exact coordinates, he had a general region that he circled on a map as areas that he caught this bycatch. And so this is really narrowing things down for us, but we're still talking about 1,400 square kilometers. So we kept seeking out information, and one of our go-to people along the coast of Labrador is Joey Undertook, who's a fishing captain and a researcher that we work with a lot. And he's from Nunavut, but when we asked him about some potential areas to look at, he said, well, he had a place that might be really good for us because he was putting out some gill nets, and he thought it was 700 meters of water, so he put out the appropriate amount of rope, and most of that rope didn't go to the bottom. And from that, he assumed that the charts were off, and he thought there might be a seamound of about 500 meters high in this area. And because his nets tangled so badly, he assumed that the bottom was quite rough. And luckily for us, he put this into his plotter as an area that he never wanted to return to, but he had exact coordinates of where this place was. So that excited us. And when we put his position next to Captain Bartlett's information, we saw it was only about 8 kilometers away. And then when we overlay all this data with our habitat suitability maps and bathymetry, we see that we're kind of getting into a bit of a hot zone. So the following year, we went out with drop cameras and we surveyed this area, some of these coordinates and some other areas nearby. And to our dismay, all we turned up is mud habitats. Now we found some interesting animals there, like some anemone fields and some sea pen fields, but not what we're hoping for in terms of these large Gorgonian corals. So we spent a lot of the rest of our time in this area mapping because we were pretty sure that there was something there, and it's just a matter of searching a little more broadly. So with these improved bathymetry maps, you can see the difference of JEPCO data on the left, which is before, and then after with our more precise multi-beam area that was shown in the red. And there was a lot of instances of deviations greater than 100 meters. So our JEPCO maps are definitely not perfect. When we process this data a little bit more, we could find areas of really high bathymetric relief. And you can see the areas in red are pretty high, but the areas in black are very steep slopes. And it's probably hard to see, but that orange circle there indicates one of these slopes that we wanted to survey. So here's the 3D rendering of what that looked like. You can see the pink and yellow dots represent areas that we visited with drop cameras and found only mud. So we were targeting these areas the next year with a brand new ROV that we had. So these cliff faces that we'd only be able to survey with an ROV that could look sideways. And much to the delight in the ROV room, they came across these beautiful cliffs of corals. I'm just going to play a little video for you here. And the interesting thing about these corals is that they typically grow like trees at the continental shelf break, pointing upwards. And in these cliffs in Makobik, they have this downward facing morphology, and they represent the closest examples of Promnoa to the coast of anywhere in Eastern Canada. So this is a pretty exciting find for us. And I'm just gonna, okay. So the interesting thing about this is with this new information of where these corals occurred, we're able to find other walls of corals nearby in Makobik Canyon Gardens, but also over 120 kilometers away at a place called the Sentinel. And we just caught a glimpse of this with the drop camera. And so we're going to spend some time this year surveying these places a little bit better and doing some more ecological studies. So stay tuned for that. And I'll just point out one other place that didn't have any large Gorgonians, but again, Joey Agnetok gave us some coordinates of a place that he knew about that had high densities. And again, even though there's no large Gorgonians, this is another place that might be of conservation value just because you can see the amount of productivity. So schools of krill and cod in the water and an abundant epifauna on the rocks down below. And so these are the kind of things that we can benefit a lot from local knowledge. All right, so we've talked about characterizing habitats. We've talked about identifying areas of high conservation value. But what about where do we put these MPAs? And so we want to make sure that we're representing these high conservation value areas and also being representative. But there's also a requirement for us to minimize negative economic and social impacts. And so this is work that's largely done by another group in DFO called Marine Protection and Conservation, and they use a tool called MarkSand to simulate different protected areas that both meet biological and ecological targets but also not put too much of a burden on the on economic and social interests. And so through this, they can lock in certain areas. You can see down here, some of the existing MPAs are locked in these simulations, and then they start looking at trying to meet objectives of representativity and important habitats. So these are completely hypothetical scenarios where we're looking at the best cost benefit in terms of conservation and with reduced costs. And a more amenable solution here is aggregate some of these specks of habitats into more easily protected areas. So really this network hasn't moved too far, but we've put a lot of input in terms of defining a network that would be representative and could be used as some guidelines for what would be useful to protect 30% of these habitats. Okay. All right, so when we do have areas recommended for protection, of course, it has to go through a stage of consultation. And so what we're seeing is a close-up view of the Laurentian Channel area, and the dashed polygons around this black line represented what the original area, which was under consideration for protection in the Laurentian Channel. So after they went out to industry and other stakeholders for consultation, that was reduced quite a bit to where the solid black line is. And so I guess one of the things that happened was they cut out a lot of the existing fishing areas, and essentially what they did was they were protecting an area that had hardly any fishing pressure, which is really great from a perspective of reducing economic impact. But one of the goals of this particular area were to protect species like dogfish, wolffish, smooth-skate, porbeagle sharks, and turtles from fishing impacts. And while this protected area doesn't really do anything to hurt those species, it doesn't do very much to move the needle because there was never any fishing in those areas to begin with. And so these conservation objectives are not maybe the best in terms of seeing positive impacts for the community. Fortunately, though, this area does have some value on its own in that it hasn't been harvested very much. And so the bottom habitats are much more intact than they may otherwise be. And as a result, they're really important habitat for sea pen fields. And so this is one of the new conservation objectives. And we wrote a paper with Corey Morris led, maybe arguing that some of the conservation objectives that are existing are maybe not the best ones for this particular area, but things like sea pens still are very valid and useful. So once we have these MPAs established, a big thing that we think about a lot is how can we track the health of these MPAs or other protected areas without destroying them? So the vast majority of our protected areas protecting sensitive benthic habitats, like these coral and sponge fields you can see in the top left. And we have a bit of a paradox is that our primary method that we use at DFO for monitoring our ecosystems, at least on the shelf, are these bottom trawls, which do significant damage to these types of habitats. And you can see on the bottom left, the potential for how much damage these trawls can do. And if we look over on the right, you can see the trawl scars left in a more open sandy bottom, probably a less vulnerable type of habitat for trawling. But you can see how much pressure is on the bottom by the trawl doors. And so a lot of the tools that we are looking at using relate to imaging technology. And so I've already talked about beta cameras and drop cameras. I've touched on our use of ROVs, but another tool we're excited about are some of these autonomous underwater vehicles like this Hydrus AUV, which is quite small and we hope will be quite powerful. We're trying really hard to buy one this year, and we just got snarled up with our procurement processes, but we hope to have some of these next year. And essentially, these can go down to 1,000 meters and follow precise transects where they take imagery. They're capable of doing some artificial intelligence identification of taxa, and going to be really useful tools for helping us keep track of what's going on in these ecosystems. So with all these imaging tools that we have a challenge that we're going to have to process all this data. And so we're trying to get some help to do that using machine learning. And so this is being done with other partners in DFO like Corey Morris's team and the National Research Council. And so we need to train some models to be able to identify a lot of these specimens. So the process really involves a human to go identify some of the taxa that we know, and then we understand what the characteristics look like on video, and then the machine learning is able to identify these things. And so if you look at this image here, you can see that we've got human annotation indicated in green, and the blue annotations represent what the machine learning has identified species as. And so you can see that we're able to detect species like starfish, anemones, sponges, with both methods. And then the machine learning also was able to detect a polychaete on its own in the bottom corner here. And so we're pretty excited about that. I'm just going to show you a quick video of what this looks like with beta cameras from some Corey Morris's work. But the green boxes are tracking the presence of cod, and you can see different cod appearing on the screen. We also have a yellow box showing crab. And if you're sharp-eyed, you might see some other more pelagic species swimming around the background that are not detected. And those are species that we've not trained the model yet for. So there's a bit of work to get all the species, but right now we're focusing on priority ones. So we're also using some newer tools that have nothing to do with cameras, and one of them is environmental DNA. And so environmental DNA is basically collecting DNA from the water or sediment, and the origins of the DNA has been sloughed off or excreted from fish or other organisms that live in this environment. And then we send the samples to a lab, they extract the DNA, and they look at the sequences and match it up to existing databases so that they can give us a list of what species exist there. Of course, there's always questions on how well this works, particularly in open ocean environments. And so we're using the benefit of our co-located sampling on the Amundsen to validate whether or not this works very well. And so in this picture here, you can see the red dots represent depth zones that we collected eDNA from, and we have corresponding data from vertical multi-net samplers that can look at all these different depths, and then oblique midwater trawls, which we can tow through that deep scattering layer to get the bulk of what would be in that particular zone. And the idea is we're interested in, does eDNA capture the species that are occurring in a larger area, like a regional area like we would have for a protected area? But can it also distinguish communities across different habitats, what would be beta diversity? And then at the smallest scale at a particular sample, how does it compare with another device? And just to give you a brief set of results, what we found is eDNA does really well detecting species across regions. So the gamma diversity is much better at detecting gamma diversity than any of the nets that we use. And it's also more than capable of detecting different communities across habitats that may only be separated by a few hundred meters. So it's able to detect differences from the surface to a little further down in the pelagic zone. But it had the downside that in cases when we're looking at alpha diversity at a single site, some of the species that nets are good at detecting, we may have detected a species there, but not picked it up in eDNA. And we think that this may be largely a function of sampling volume, where the nets are sampling sometimes up to millions of liters of water, where our eDNA samples are only looking at about 5 liters of water per replicate. And of course, if we have such rich data like eDNA, we can really fill out some important things like food webs, which we might want to use as indicators. And so you can see the eDNA-based food web on the left compared to a pelagic trawl food web on the right. And so, we're hopeful that these will be important tools for monitoring processes in our systems. And of course, when we come up with these indicators, we also have some pressure to make sure that these indicators can reliably detect change. If things start going bad in these protected areas, we need to know about it so we actually have a chance to actively manage it. And so one of the tools we're applying where we can is power analysis. And so the power analysis tells us what the likelihood is of us being able to detect change if it occurs. And so we use real data to inform simulations, and we looked at conservation objective species in the Laurentian Channel using trawl data, which we have long-term data sets for in this area. And unfortunately, what we found was the trawls they're probably very good for what they're designed for managing fisheries, but for tracking the abundance of particular species that are variable and maybe not too abundant, they don't work very well. And so at very best, we found we could only detect very huge declines in some species if we increase the amount of trawling considerably. So right now, we do about 23 trawls a year in these protected areas, and to get any semblance of power, sometimes we would have to increase it to 100. So that's economically infeasible for our programs and also not desirable because of the potential damage trawls could do to these areas. In contrast, we found that some other metrics, the community metrics like richness, might be much more statistically powerful if we apply those. And so we're doing a lot of work trying to figure out which indicators are going to work in the long term. We also are concerned about these protected areas that are static in nature. They're fixed in space. And we know that our communities are very likely to change over time, especially with the forces of climate change. So in this particular case, what we're doing is using climate projections and looking at different climate scenarios. Seems like Siri is trying to jump in on here. 1 second.
Something went wrong.
All right. Sorry about that.
I'm sorry.
I'll just move that. Okay, so we're using these climate models and we're combining it with physiological models to understand where these species are likely to go. And if we look at the plots on the right, these are 1% growth contours for different species of cod, Atlantic cod, rock cod, and Arctic cod. And you can see the historical data from 1981 to 2005 up here. And of course, Arctic cod growth is further north, as you might expect, and Atlantic cod is the furthest south. If we go down to the bottom right corner under the most extreme scenarios, we can see that these distributions are shifting quite a bit farther north. And we can extend that to a protected area perspective and include some additional environmental variables like oxygen and chlorophyll A, along with life stage specific variables. And that allows us to get an understanding of what habitats will be suitable. And so this paper here, we're looking exclusively at Atlantic cod. And we find that the habitat is declining. You can see the red zones in this map in the Gulf and Southern Grand Banks, whereas habitat conditions are actually improving overall further north. And in some of the areas like Funk Island Deep and Hawk Channel, which are Atlantic cod as conservation species, or conservation objective species, sorry, those areas should continue to be good going forward. But if we contrast this with the Laurentian Channel, which we see on the right, we can see that over time, egg survival is still going to be strong, even with things heating up. Juvenile growth, which is the next panel down, is going to be declining a little bit, except for the most extreme scenarios. But it's really the aerobic growth index, which represents the habitat suitability for the female spawners, is an area where it's going to tank out about mid-century. And we can see that there's virtually no habitat suitable for these female spawners after about 2050. So we can use these tools and apply it for different species across our protected areas and get an idea of how they're going to change over time, and maybe what we might need to be mindful of when we're establishing monitoring programs. So with all this talk of change, of course, it's really important that we build programs that we can adaptively manage. And so this is one thing we're not really strong on right now in general from a management perspective. But I'll give you an example here. The Hopedale Saddle protected area, again, was put in place to protect sensitive benthic habitats like corals and sponges. And when we've been doing our monitoring, we've been finding many instances of this creature, which is also very old, or can be very old, and can be very sensitive because it's old. So these Greenland sharks seem to be at very high densities in this area. And so we're implementing a tagging program this summer with satellite tags to see if they actually stick around in these areas long enough that they could be protected. And if so, we could propose these species as another conservation objective species to go along with these coral and sponge areas that are already designated for. And similarly, adaptive management can be really important for areas that are already in existence for conservation species that are already listed. So the Gilbert Bay Marine Protected Area was the first one implemented in our region back in 2004, 2005. And we can see from the top plot that there was a a strong decline over the last 20 years or so in golden cod. And so the golden cod are subspecies of cod that are starting to disappear. And so even though we've designated this NPA, the protections are not working well enough to preserve the stock. And the thinking is that the species is migrating in and out of the protected area into the coastal zone, where it's mixing with offshore Atlantic cod and being captured in the fishery. And so to be able to protect the species, we need to have the tools to be able to adaptively manage, which is something that's challenging. So the last general thing I'm going to talk about is sustaining these programs. And so in general, we know that funding comes and goes. It goes through different waves. And for programs to be useful, they need to survive in the long term. And so we really are mindful that we need to have a program that's valued and cost efficient. So some ways that we make it cost efficient is by making sure we're co-locating our sampling so we can get lots of different types of data at the same time, that they're complementary. And we also want tools that we can use off smaller vessels. And if you look at the picture down below, this is the Amundsen visiting Nain, and you can see the red boat, but we also see some smaller vessels in the near ground that can be useful for many of the tools that we like to implement, and they're much less expensive than the Amundsen. Another important part is having useful indicators. And so I think our RV survey program on the shelf is a great example of that the data is used on a yearly basis for guiding industry. And so if that were to ever stop, there would be huge calls to implement the programs again. And so that certainly makes politicians less inclined to make these cuts. And the last part, of course, is the general community involvement. And so again, it's really important that we make time to make the average Canadian value these areas. And so this public engagement, ideally, we're bringing people out to these protected areas. And so you can see in this top picture, one of our team, Jess here, is presenting to a group of Innu in coastal Labrador on a Students on Ice program. But the reality is that many of these areas are remote and it's difficult to bring people out there. So we kind of have an obligation to bring protected areas to the people. And we do that through community meetings, but also through some other approaches, like, for example, our benthic ecology team at DFO curated a collection of corals that you can see in that middle picture down here that they donated to the Nunavut government that they could use for education purposes, and also just let people know what's living in their waters and help generate a sense of pride of what belonged to them. And another means of doing this is, of course, trying to do our best to be active in the media. And so that can be done through social media, but also, we try to be involved in where we can in television and radio and even print media. And so this is a great opportunity for us to to profile some of our partners. And on the right, you can see Joey Agnetuk, and he was featured in an article that talked about his role in discovering the Makova Canyon Gardens. So to close and summarize all this, I'd just like to go over the fact that we have really intense timelines that we have to make. This 30 by 30 timeline is going to be really challenging, and to have any chance of making it, we really need to work together. So that means across science disciplines, with different knowledge holders and with industries, everybody providing information so that we can have the best information for these decisions. We also have to make sure that our protected area network is representative, but also capturing these sensitive habitats. And again, that relies heavily on more information than we have right now. And of course, a big part of this is implementing these protected areas in an equitable manner. So that means when we're doing this research, that we're inclusive of different groups, and it will require lots of consultation and collaboration. And when these places are implemented, co-management. And in my experience, the co-management really seems to be an effective solution. And we also need to acknowledge that we're not going to get everything right the first time. So we know our environment's changing. We know our knowledge is improving all the time, and we need to build in mechanisms for adaptive management. So this is something I think that we need to change some policy to make work better. And then lastly, I think as scientists, we can get caught up in writing papers and just the pure science aspects, but it's really important that we leave time to make sure that Canadians are aware of how valuable these areas are, and so that they support these initiatives in the future. And so with that, I think I'm all done. Thanks for listening, and I'm happy to take some questions.
Thank you, Dave. That was awesome. And thank you for sharing some of those videos. They're very neat to see. I'll open it up to the floor, and if anyone is online, please raise your hand and I can unmute you. Check it around.
Go first.
Yeah, Rachel.
Hi, Dave. It's Rachel here. I'm going to see you through the camera. Awesome presentation. It was really cool to see all your work put together in one presentation. I had a question about eDNA. So are you incorporating current speed and directionality for your eDNA data to see like if it's coming from a certain area rather than focusing on one spot?
Actually, we've not done that. And to be honest, given from the results that we've had so far, I haven't really seen a big purpose. We've tried to do some trials with in collaboration with the lab, where we put some caged fish out in an open ocean environment, and we've seen how far we can detect these. The results are not complete yet. So we're hoping to have some more detailed information on what currents might look like, because we can sample on the downstream side of those cages, and we can also sample on the upstream side. So in the open ocean environments, I think there's probably some lateral movement, but the areas seem to be relative homogeneous out there. But a lot of studies do show in coastal areas that they're detecting latitude or lateral changes in communities within scales of hundreds of meters, actually. So, so we haven't put a huge amount of time into that, but we are hoping to get some interesting results from that cage study that we're doing.
Thank you.
Yeah, John.
Hi, Dave. John here. And I had a question about mapping. So you talked about multi-beam mapping, and you talked about the Challenger expeditions way back when. And we have these, we're at the Marine Institute here, and some groups do sort of exclusively bottom mapping. Some groups might do fish and invertebrate sampling. Just wonder if you could comment on how much you think mapping has to lead those other collections to inform sort of site designation versus mapping being just another component of data collection in these areas.
Yeah, good question. I think based on my experience, when we've gone out blind without any maps, bottom maps to do our work from, we've come home empty-handed a lot more than if we have detailed maps. And so, you know, I think we've had other groups that would ask their recommendations on how we should go, how they should go forward. And we strongly recommend spending a year just mapping the area and figuring that out first. But, you know, some groups at MI that I've worked with, like Zach McMillan, Kenny's work, I mean, he's been lucky. He's been able to combine those bottom maps with the fish communities in the same project. And so that's kind of a nice way to do it. And sometimes that's been done retrospectively in the case of some of our telemetry data that we've donated. But in general, yeah, if you're looking for those rare habitats, I think we need to go more than just go out there blind and just hope that we're going to hit on something important.
Yes.
Yes. Sorry.
Hi Dave, I'm Laura, working with Maxime Lepoix here in CIPR. Thank you so much for the presentation, it was so interesting. And I have a question related to already existing marine area. You talked about the seapen conservation. I was wondering if there is, do you manage, how are managed the existing areas, and how do we see if there are some changes, if it's efficient, for example? Yeah, I don't know if it's clear.
Okay, nice to meet you in person, Laura, first of all. We've communicated a lot. So if I understood the question correctly, you're wondering how we can track the changes of sea pens in the Laurentian Channel, is that correct?
Yeah, for example, or any other changes, like is there a way to check if the marine protected area has a concrete effect on the where it is?
Right. So the tools that we're using for the sea pens are drop cameras. And so we're hopeful that AUV that I mentioned earlier will be able to do precise transects again and again, so we can actually count the same sea pens to see if they're changing. But really the challenge with that protected area is that where there's no fishing impact, we're really looking to see if there's change within the area as opposed to comparison inside, outside. Because the way it was set up is very much biased. There's no corresponding area outside that is not fished, or vice versa, the areas that used to be fished inside. So we're really focusing on areas inside the area, and we're looking for declines and basically preserving what we have as opposed to seeing it recover, because they were never damaged in the first place. And that's a good question, because I think there's a lot of pressure on protected areas to show improvements in ecosystems. But if we're trying to set them up with while minimizing economic impact, we're biasing these areas to areas that were never fished to begin with. And so it kind of puts us in a tough position. And it's really a matter of awareness that we should manage our expectations accordingly.
Thank you.
Hey, Dave. I don't see any questions online, but I got one final question So, and I guess in regards to adaptive management, I was wondering what your opinion would be on more of like an EBM approach, like a ecosystem-based management approach for these marine conservation and protected areas. Do you think it would be better than like a specific species of interest, or do you think a more holistic approach would be better?
Yeah, I mean, I think I'm in favor of that generally in terms of indicators and conservation objectives. One of the things about relying on individual species is there's a strong potential that they could, with climate change, get pushed out of that protected area. And then we're left with a situation where that protected area has no value by the criteria that we originally defined for that. So if we can look at processes, it doesn't matter if different species move in or not, we still have a potentially thriving ecosystem of new species, but it's still providing ecological value. And so, yeah, I would be very much in favor of some of those ecosystem-based approaches.
Perfect, thank you. Yeah, I wholeheartedly agree. Thank you, Dave, and thank you for everyone who joined in person, and thank you for everybody who joined online. Our next seminar will be next Wednesday, featuring Dr. Neil Bowes, the Memorial University of Newfoundland president. We'll host a seminar here in this lecture hall, room W3005, and we'll be live streamed via WebEx for those who cannot make it. So we hope to see you then. Perfect. Thank you, everybody.
Thanks, everyone.